A very good morning to one and all. Dear students, today we are going to study about the factors affecting drug absorption. In previous lecture we studied about what are the different mechanisms of drug absorption. In that we studied various mechanisms like passive diffusion, active drug transport, endocytosis and many more. Now in today's lecture we are going to study about which factors mainly affect the process of drug absorption. The lecture the lectures for factors affecting drug absorption are mainly divided into two parts. So today we are going to cover part 1. The contents of my today's presentation are introduction to absorption, then pharmaceutical factors, physicochemical properties of drug substances and formulation factors. So these factors we are going to cover in today's presentation or today's lecture. Introduction. The chain of events that occur after administration of solid dosage form such as tablet or capsule until its absorption is as follows. Means which steps mainly occur before the drug absorption, as most of the drugs are administered in the form of tablets as well as capsule. Here we are seeing the example of such solid dosage forms. After administration of solid dosage form, either a tablet or capsule, consider firstly we will say about the tablet. It mainly disintegrates, then its aggregates are formed, then after deaggregation its fine particles are mainly formed and they are mainly dissolved into the aqueous GI fluid. In case of capsule, its shell firstly dissolves, then the drug particles are mainly dissolved into the aqueous GI fluid. So these steps like disintegration, deaggregation mainly and dissolution mainly takes place before the drug absorption. After dissolution, so drug is there into the aqueous GI solution. As we already discussed that drug must have sufficient hydrophilicity in order to be dissolved into the aqueous GI fluid, then it will be ready to cross the cellular membrane. Okay, so after the drug dissolution, drug is there into the solution at the absorption site. Then after that the non-ionic form of the drug easily crosses the gastrointestinal barrier or cellular membrane, then that goes into the systemic circulation or into the blood. Here the ionic drug cannot cross the GI membrane, as we have already discussed in the previous lecture that only non-ionic drug or unionized drug molecules can cross the gastrointestinal barrier. Okay, so these are the series of steps which mainly occur before absorption as well as during the absorption. So the factors affecting drug absorption are mainly divided into two types, that is A, pharmaceutical factors, and patient related factors or physiological factors. The pharmaceutical factors are mainly divided again into two categories, that is physicochemical properties of drug substances and dosage form factors. Various physicochemical properties of drug substances are drug solubility and dissolution rate, particle size and effective surface area, polymorphism and amorphism, solvates and hydrates, salt form of drug, and drug pKa and lipophilicity and gastrointestinal pH. So these are the physicochemical properties of drug which affect the absorption. The next category of pharmaceutical factors are dosage form factors. Those are disintegration type, manufacturing variables, nature and type of dosage form, pharmaceutical ingredients, product age and storage conditions. Various patient related factors are age, gastric emptying, intestinal transit, disease states, blood flow to the GIT and pre-systemic metabolism. So this is the enlisted factors which mainly affect the drug absorption. So first factor means the first physicochemical property of the drug is drug solubility and its dissolution rate. Here into the figure we can see that there are two rate determining steps into the absorption of drug from orally administered formulations. Those steps are rate of dissolution and second rate of drug permeation through the biomembrane. As we already discussed that solid dosage form firstly disintegrates, then its drug particles are produced and those drug particles are dissolved into the aqueous GI fluid. Okay, but the process of dissolution is the rate determining step for lipophilic drugs. Hydrophilic drugs easily dissolved into the aqueous GI fluid, so dissolution is not problem for such a kind of drugs. But lipophilic drugs do not dissolve very easily into the aqueous GI fluid, and for them the rate determining step or the step which occurs slowly is the dissolution. While when drug is there into the solution at absorption site, it has to permeate throughout the biomembrane, and this permeation is the rate determining step for hydrophilic drugs, and after permeation drug is there into the body. Okay, so these two steps, that is dissolution as well as permeation, play a very important role into the absorption of the drug, and dissolution and permeation means hydrophilicity and lipophilicity are the characteristic of drug itself. So based upon this permeability and solubility of drug, Amidon and his co-workers or colleagues developed a special system, that is BCS, means Biopharmaceutical Classification System, which classifies the drugs into 4 categories or 4 classes. Those are class 1, class 2, class 3 and class 4. So class 1 drugs are having high solubility as well as high permeability. So there is no problem in case of absorption of drug. These kind of drugs are well absorbed into the systemic circulation. Example is diltiazem. Then class 2 drugs are having low solubility as well as high permeability. Instead of having high permeability, they are having low solubility, and that's why such a kind of drug have variable absorption pattern. So that mainly depends upon the structure of the drug. For example, nifedipine. Okay, class 3 drugs are having high solubility but low permeability, and that's why they are also having the variable absorption pattern. For example, insulin. The class 4 drugs are having low solubility as well as low permeability, and that's why these kind of drugs are poorly absorbed. Example is Taxol. So from this classification we come to know that the class 1 drugs which are having high solubility and permeability, while class 3 drugs which are having high solubility but low permeability do not have that much of absorption problem. While the class 2, that is low solubility, and class 4, that is low solubility and low permeability, also show poor absorption. And that's why class 4 drugs are generally administered by the intestine, by the IV route generally. So an important requirement for the absorption of drug is that it must be present in aqueous solution. In previous lecture we have already studied that sufficient amount of drug must be dissolved into the aqueous GI fluid, and this mainly depends upon its solubility as well as dissolution rate. That's why these two factors are very important into the absorption of the drug. So dissolution is a process in which solid substance solubilizes in a given solvent. Means the drug particle mainly dissolves into the aqueous GI fluid. So that process is mainly called as the dissolution. There are various theories which mainly explain the process of dissolution. We are going to cover 2 theories, that is diffusion layer model as well as surface renewal theory. So dissolution is a process in which solid substance solubilizes in a given solvent, that is the mass transfer from solid surface to the liquid phase. So first theory is the diffusion layer model or film theory, which is the simplest one as well as most common and widely accepted. The process of, according to this theory, the process of dissolution mainly consists of two consecutive steps. First, there is solution of solid. In this step, the solution of solid is mainly there to form a thin film or layer at the solid liquid interface, and which is called as stagnant film or diffusion layer, which is saturated with the drug. And this step is usually rapid, while the second step is diffusion of soluble solute from the stagnant layer to the bulk of the solution, and this step is slower and is therefore the rate determining step in drug dissolution. Here into the image, into the figure, you can see these two steps. First, there are mainly four components, that is dissolving drug, dissolving solid drug, then GI lumen, GI barrier, and the blood. Okay, so when solid drug particle starts to dissolve, the solid liquid interface is mainly formed, and surrounding the solid liquid interface there is formation of stagnant layer, or you can say there is formation of a film, and throughout that film the diffusion of molecule mainly takes place. So this series of steps mainly occur according to the diffusion layer model as well as film theory. So the modified Noyes and Whitney equation mainly explains the rate of dissolution, or we can say diffusion of molecules from this kind of stagnant layer. Okay, so the equation is dC upon dt is equal to dAk W by O Cs minus Cb upon Vh. Okay, so here dC upon dt is the rate of dissolution, then d diffusion coefficient. The greater the value of d, the faster will be the dissolution of the drug. Okay, then A is the surface area of solid. Greater the surface area, faster is the drug dissolution. Then next component is Kw by O, that is water oil partition coefficient. Higher is the value, more the coefficient of drug and its hydrophilicity, and therefore faster is the dissolution of drug into the aqueous GI fluid. The next important factor is Cs minus Cb. Here into the figure you can see there are mainly two concentration terms, that is Cs and Cb. That is, Cs mainly stands for concentration of drug into the diffusion layer, while Cb means concentration of drug into the bulk of the solution, that is aqueous GI fluid, and this concentration difference is mainly called as concentration gradient. So greater the concentration gradient, faster is the diffusion and drug dissolution. So this factor is particularly important. The next factor is V, that is the thickness of stagnant layer. More is the thickness, lesser the diffusion layer, and that's why less is the drug dissolution, and this can be increased by increasing the agitation. Okay, so these factors are mainly according to the modified Noyes and Whitney equation, which mainly explain the process of dissolution. Okay, so likewise solubility and dissolution rate mainly affect the process of drug absorption at very larger extent. Next, second theory of drug dissolution is Danckwert's model or penetration or surface renewal theory. It is called as Danckwert's model because it was discovered by Danckwerts. So Danckwerts takes into account the eddies or packets that are present into the agitated fluid, which reach the solid liquid interface, absorb the solute by dissolution and carry it into the bulk of the solution. Okay, so according to this theory, there is formation of a packets of the solvent. Here into the figure you can see there are mainly two kinds of packets which are of fresh solvent, and there are some of the packets which carries the drug into it. Okay, so these packets can be called as the eddies. So these packets are mainly produced into the solvent, and these packets mainly travel towards the dissolving solid or at the solid liquid interface, and the drug is mainly loaded into those packets, and again those loaded packets are transferred or they are mainly carried into the bulk of the solution, and likewise dissolution is mainly carried out. Okay, so these packets get continuously replaced by new ones and exposed to new solid surface each time. Thus the theory is mainly called as surface renewal theory. Okay, so these are mainly two theories which mainly explain the process of dissolution. This is about the effect of drug solubility as well as dissolution rate onto the drug absorption. Now the next factor is particle size and effective surface area. The smaller the particle size, greater is the effective surface area, then more intimate contact will be there between solid surface and aqueous solvent, higher is the dissolution rate and increase in absorption efficiency. Okay, so particle size and surface area of solid surface are inversely related to each other. Smaller the drug particle, greater is the surface area, and if greater is the surface area, then more will be the dissolution, and if dissolution is more, the rate of absorption will also be higher. There are mainly two types of surface area. Those are absolute surface area and effective surface area. First, absolute surface area, which is the total area of solid surface of any particle, and effective surface area, which is the area of solid surface exposed to the dissolution medium. Since surface area increases with decreasing particle size, decrease in particle size will result in higher dissolution rates. However, it is important to note that it is not the absolute surface area but effective surface area that is proportional to the dissolution dissolution rate. Greater the effective surface area, we already discussed that greater the effective surface area, more intimate the contact between the solid surface and aqueous solvent, and faster is the dissolution of the drug. And that's why micronization is one of the size reduction technique which is mainly used to increase the effective surface area. There are a number of drugs which are poorly absorbed, but after the micronization their absorption is mainly increased at the larger extent. For example, the bis-hydroxycoumarin, digoxin, griseofulvin, nitrofurantoin and 12-butamide, as well as many more drugs. So griseofulvin has extremely low aqueous solubility and material of normal particle size gave rise to poor and erratic absorption. Micro-sized particles improve absorption, but it is improved even more when it is formulated in ultra micro-sized particles as a molecular dispersion in polyethylene glycol. So number of efforts are mainly taken in order to increase the particle size and thereby in order to increase the absorption rate of the drug. Next factor is crystalline as well as amorphous form. When a substance exists in more than one crystalline form, those different forms are designated as polymorphism, and the phenomenon those forms are designated as polymorphs, and the phenomenon is mainly called as polymorphism. These polymorphs may have different physical properties such as dissolution rate as well as solubility. Depending on their relative solubility, those polymorphs or crystalline forms, we can say, can be divided into two categories, that is stable form and metastable form. The stable form represents the lowest energy state, highest melting point, least aqueous solubility and dissolution rate limited. Here we can see that stable form are having low aqueous solubility as well as low dissolution rate. But in case of metastable form, which are having higher aqueous solubility as well as better absorption and better bioavailability, and that's why metastable form of the drugs are mainly preferred into the formulation of different dosage forms. For example, chloramphenicol palmitate exists in three polymorphic forms, that is A, B and C. In that, B form shows best availability while form A is virtually inactive biologically. Another example is riboflavin. The polymorphic form 3 of riboflavin is 20 times more water soluble than form 1. Okay, only 10% of pharmaceuticals are present in their metastable forms. Okay, so likewise the metastable form are mainly preferred into the formulation. Some drugs can exist in amorphous form, that is having no internal crystalline structure. Such drugs represent highest energy state and can be considered as supercooled liquids. They have greater aqueous solubility than crystalline forms because the energy required to transfer a molecule from crystal lattice is greater than that required for non-crystalline or amorphous form. For example, amorphous form of novobiocin is 10 times more soluble than crystalline form. Okay, so here we can see the difference between crystalline form as well as amorphous form. Crystalline form is less soluble, slower dissolving and not absorbed to significant extent, while the amorphous form is more soluble, rapidly dissolving and readily absorbed. And thus the order for dissolution of different solid forms of drugs is, first one is amorphous, which is having highest order of dissolution, then after that metastable form, and then the stable form. Okay, so depending upon this behavior, the suitable form of that particular drug is mainly preferred into the formulation, so that there will be no problem into the absorption or dissolution of the drug. Then next factor is solvates or hydrates, means hydrous as well as anhydrous form of the drug. Solvates means when drug is in association with any other solvent, that is mainly called as solvate. When the solvent in association with drug is water, then it is mainly called as hydrates. Generally, the anhydrous form of drug has greater aqueous solubility than hydrous form. This is because the hydrates are already in interaction with water and therefore have less energy for crystal breakup in comparison to anhydrates for further interaction with water. For example, the anhydrous form of theophylline and ampicillin have higher aqueous solubilities. They dissolve at higher rate and show better bioavailability in comparison to their monohydrate and trihydrate forms respectively. Next factor is salt form of the drug. Most of the drugs are either weak acid or weak bases. So at a given pH, the solubility of drug, whether acidic or basic or its salt, is a constant. While considering the salt form of drug, pH of diffusion layer is important, not the pH of bulk of the solution. Into the diffusion layer theory we have studied, there is formation of a layer surrounding solid liquid interface, and that layer is mainly called as diffusion layer. So its pH is mainly important while considering the salt form of the drug. There are many more drugs which are available into their salt form. The drugs are converted into the salt form because in order to increase the dissolution rate of the drug and in order to increase the absorption rate of the drug. There are many more examples. For example, diclofenac sodium, metformin hydrochloride, tetracycline hydrochloride, tetracycline sulfate, omeprazole magnesium. So all these drugs are mainly converted into their salt form because in order to increase the dissolution rate. For example, consider the salt of weak acid, which increases the pH of diffusion layer, which promotes the solubility and dissolution of weak acid, and absorption is bound to be rapid. Here, from this sentence we come to know that ionization favors the dissolution. Means salt of weak acid means strong base will be its salt. For example, strong base means Generally, with weakly acidic drugs, a strong base salt is prepared, such as sodium and potassium, while in case of weakly acidic drugs, a strong acid salt is prepared, like hydrochloride or sulfate salts. Okay, so take the example of diffusion layer. So salt of weak acid, if it is combined with strong base salt, then what will happen? The pH of diffusion layer will increase, that it may go up to the pH 5 to 6, and thereby, and after the ionization, drugs are completely dissolved into the aqueous GI fluid. Okay, so likewise salt form of the drugs is also one of the another approach in order to increase the dissolution rate of the drug, and thereby in order to increase its bioavailability. So here into the figure we can see this is the example of salt of weak acid, which is diffusing out throughout the diffusion layer after combining the salt of weak acid with its strong base. Definitely its pH increases, and due to lower pH of bulk of the solution, which is 1 to 3, the diffusion of soluble drug particles mainly takes place, and there is rapid dissolution. Drug will be there into the solution and it will be absorbed into the systemic circulation. Okay, in case of salt of weak base, we will have to use its strong acid salt. Then what will happen? The pH of diffusion layer will be lower, it may be up to 1 to 3. Then into the bulk of the solution, its dissolution will increase and its rate of absorption will also increase. That's why the principle of in situ salt formation has been utilized to enhance the dissolution and absorption rate of certain drugs. For example, aspirin. Aspirin is firstly dissolved into the dissolved into the water. Then its tablets are mainly called as buffered alkaline tablets. And due to in situ salt formation, there is no problem into its absorption. Okay. Next factor is drugs pKa, its lipophilicity and gastrointestinal pH, and all these factors are mainly explained by pH partition hypothesis or pH partition theory, which states that for drug compounds of molecular weight more than 100, which are primarily transported across the biomembrane by the process of passive diffusion, the process of absorption is governed by pKa of drug, which determines the degree of ionization at particular pH, pH at absorption site, and lipophilicity of an ionized drug. Means according to pH partition hypothesis, the process of passive diffusion or the process of drug absorption is mainly governed by three factors. First, pKa of drug, then pH at the absorption site, and lipophilicity of unionized drug. Here, ideally for optimum absorption, drugs should have sufficient aqueous solubility to dissolve in aqueous GI fluid and lipophilicity high enough to permeate across biomembrane and into the systemic circulation. So, the next sentence is very important, that a perfect hydrophilic lipophilic balance should be there into the structure of drug for optimum bioavailability. Here the drug pKa and gastrointestinal pH plays important role. The amount of drug that exists in unionized form is a function of dissociation constant, that is pKa of drug and pH of fluid at the absorption site. It is important to express the dissociation constant, that is pKa, of both acidic and basic drugs. The lower the value of pKa of acidic drug, stronger is the acid, that is greater the proportion of ionized form at particular pH. The higher the pKa of basic drug, the stronger the base, and thus from the knowledge of pKa of drug and pH at the absorption site, the relative amount of ionized and unionized drug in particular pH can be determined. Okay, so here into the table we can see if drug is having drug is very weakly acidic, whose pK is less than 8, then it will remain unionized at all pH values, and therefore it will be absorbed along the entire length of GI tract. Here, one of the important point to note is that for a drug permeation, drug must be there into its unionized form, and that's why very weak acid which remain unionized at all the pH values, and therefore these kind of drugs are mainly absorbed from stomach as well as small intestine as well as large intestine. There is no specific site for such kind of drugs for their absorption. Next one is moderately weak acids, means unionized in gastric pH, that is the stomach pH. The pH of the stomach is about 1 to 3. So into the stomach it will remain unionized. Okay, while it will be ionized into the intestinal pH because the pH of intestine is greater than 5 to 6, and that's why it will be ionized into the intestine, and therefore such a kind of drugs are better absorbed from the stomach. Okay, next one is stronger acids whose pK value is less than 2. Such a kind of stronger acids remain ionized at all pH values, and that's why they are poorly absorbed from the GI tract. In case of very weak bases whose pH is less than 5, they remain unionized at all pH values and absorbed along entire GIT. Moderately weak bases, they remain ionized at gastric pH but unionized at intestinal pH, so they are better absorbed from the intestine. Okay, and stronger bases which pK is greater than 11, they remain ionized at all pH values, and that's why they are mainly poorly absorbed from the GIT. From the table we come to know stronger acids as well as stronger bases, means there will be stronger ionization, more ionic form of the drug will be there, and that's why they remain ionized at all pH, and that's why they are mainly poorly absorbed from the GI tract. Okay, so this is the effect of drug pKa, lipophilicity and gastrointestinal pH. So here very important line or conclusion of the pH partition theory is that a drug should have sufficient aqueous solubility so that it will be dissolved into the aqueous GI fluid, and it should have sufficient lipophilicity so that it will be easily permeated throughout the cellular membrane, and that's why a perfect hydrophilic lipophilic balance should be there in the structure of the drug for optimum bioavailability. Okay, so these are the factors, these are the physico-chemical properties of the drug which mainly affect the drug absorption. Next category of factors are dosage form factors. Okay, means the factors which are related with dosage form. Previous category was the factors which are related with drug. Now this category of factors which are related with dosage form. There are many more factors which are mainly associated with dosage form. For example, first one is disintegration time. Rapid disintegration is important to have rapid absorption, so lower DT is required. So lower disintegration time is mainly required. Okay, so the tablet or capsule must be disintegrated at rapid rate so that absorption will also be higher. Now disintegration time of tablet is directly proportional to the amount of binder and compression force. Okay, so if amount of binder is mainly increased, then as well as the compression force is mainly increased, then what will happen? The hardness of tablet will also be increased, and that's why the disintegration of tablet will be very poor and disintegration time will be increased. Okay, so amount of binder and compression force plays an important role in case of disintegration. And one thing should be remembered that in vitro disintegration test gives no means of guarantee of drugs bioavailability, because if the disintegrated drug particles do not dissolve, then absorption is not possible. Okay, and that's why dissolution test is particularly important in order to evaluate the absorption efficiency of drug from its dosage form. Okay, next factor is compression force. As we already discussed, higher compression force yields a tablet with greater hardness, and therefore it reduces the wettability and hence have long disintegration time. But on the other hand, higher compression force may cause crushing of drug particles into smaller ones with higher effective surface area, which in turn decrease in disintegration time. So effect of compression force should be thoroughly studied on each formulation. Next, third factor which is related with dosage form is the nature of dosage form. Tablets, capsules, powder, suspension, emulsion and solution. The relative rate at which a drug from a dosage form is presented to the body depends upon the complexity of dosage form. The more complex a dosage form, greater the number of rate limiting steps and greater the potential for bioavailability problems or absorption problems. The rate at which a particular dosage form releases the drug following administration as is given into this image. Okay, so here we can see the tablet is very complex dosage form because the number of steps takes place before the drug absorption. For example, disintegration. After disintegration, the granules are produced. After deaggregation, fine particles are produced. Those fine particles dissolved into the aqueous GI fluid. Their drug is there into the solution, and after that drug is mainly absorbed. Okay, so disintegration, deaggregation, formation of fine particles, dissolution, drug in solution. So these are the steps which mainly takes place before drug absorption if drug is administered in the form of tablet. Then next to the tablet there is the capsule in which dissolution of shell takes place, then deaggregation, fine particles, dissolution, drug in solution and then absorption. Powders, fine particles, dissolution, then absorption. Emulsion, firstly drug dissolution and then absorption. Solution means drug is already there into the solution form, drug directly absorbed into the systemic circulation. So from this image we come to know that the rate of absorption is fastest in case of solution, while rate of absorption is lowest in case of tablets. So as we move from solution to the tablet, the rate of absorption mainly decreases. So this mainly depends upon the complexity of the dosage form. Then next factor is pharmaceutical ingredients or excipients. More the number of excipients in dosage form, more complex it is and greater the potential for absorption and bioavailability problems. We already discussed that tablet is very complex dosage form because the number of excipients within the tablet are too much more and number of series of steps mainly occur before absorption of drug from the tablet, and that's why the complex dosage form offer greater potential or problem in case of absorption as well as bioavailability. First, vehicle. The miscible vehicles, if we used, then absorption will be rapid. If immiscible vehicles we are using, then absorption mainly depends upon its partitioning from oil phase to aqueous body fluids. Okay, so that's why the vehicles used into the various liquid dosage forms are generally miscible one in order to have better absorption. Then diluents. Hydrophilic diluents form the hydrophilic coat around hydrophobic drug particles and therefore they promote the drug dissolution and absorption of poorly soluble hydrophobic drugs. Then binders and granulating agents. Generally this should also be hydrophilic one because they impart hydrophilic properties to granule surface and thereby there will be better dissolution of poorly wettable drugs. Here the concentration or amount of binder is particularly important one that we already discussed. More amount of binder increases the hardness of tablet and thereby decreases the dissolution as well as disintegration rate of the tablet, and therefore optimum concentration of binder is very essential for drug absorption. Then disintegrants, which are mostly hydrophilic in nature. Decrease in amount of disintegrants may lower the bioavailability. Okay, so amount of disintegrant is particularly important one, and which are mostly hydrophilic in nature. Lubricants, which are commonly hydrophobic in nature and therefore inhibits penetration of water into the tablet and thus dissolution and disintegration. So that's why amount of lubricants used should also be optimum one. If concentration of lubricant is too much higher, then it will inhibits the penetration of water and thereby it will create the problem in case of absorption of drugs. Then surfactants may enhance or retard drug absorption by interacting with drug or membrane or both. The surfactants mainly increases the absorption by various ways. For example, by promoting the wetting properties or dissolution of the drug, then better membrane contact of the drug for absorption and enhanced membrane permeability of the drug. Okay, so surfactants are widely used into the formulations as wetting agents, solubilizers and emulsifiers. So these were the pharmaceutical factors which mainly affect the drug absorption. In next lecture we are going to study next category of factors, that is patient related factors or physiological factors. Thank you.